disclaimer here. My background is physics and mathematics, and I just want to say phytoplankton and biology is hard, and so I apologize if I say anything that you got more expertise than me. That's fine. But the reason I'm sort of talking about this is my background was my PhD was looking at tracking trichodesmium, which is a filamentous colony-forming cyanobacteria that we see. I'll talk a little bit about it. I put it in there on purpose. So that's my background with sort of phytoplankton discrimination and classification. And then lately I've been working on implementing scientific software into NASA's sort of processing system for different phytoplankton algorithms for the PACE era. That's my background. And I want to sort of, for this, I borrowed a lot of slides from Jeremy Werdell, Ali Chase, and a lot of advice from Ivana Setnich, who's the biogeochemistry science lead for the PACE mission. So really important to thank those guys for providing materials for me here. Okay, here's a little bit of an overview of what we're going to do today. I'm going to try and get through most of it, hopefully in an hour. And then at the last half hour, I decided instead of me telling you about case studies, I'm going to give you guys some very, very quick sort of briefs. And I want you to come up about a problem that you might be able to solve with some of the things I talk about. I'm going to split you into groups, and hopefully you can come up with brainstorm some ideas and then just report back some key points. We'll see how we go, see if that works. But because there's so many screens around it, it's easy to project those up. Okay, first off, phytoplankton. Who in the room is has an interest or researching work with phytoplankton? Can I see? Okay, good. Everybody. So everybody's going to tell me I'm stupid when I make a mistake. That's all right. So I don't really have to, this is great. But for those of you who aren't phytoplankton people, you're like me, your background's in physics. Phytoplankton, microscopic algae. They live in aquatic ecosystems. So we've probably got some freshwater researchers in the room. So I'm going to make sure I don't forget them. But in the oceanographic community, we've been studying phytoplankton for a long time. And this is just an example here, looking underneath a microscope, just to show you phytoplankton come in all different shapes and sizes and colors. We like to say size, morphology, and color. And yeah, just to give you an idea, I think I just did like a web search and tried to make a word cloud. This is something new to me, just to see what popped out when you search for phytoplankton community composition. I got these sort of things coming out: diatoms, nutrients, cells, species. So just to give you an idea, this is the hot topic words, so to speak, and some of these words will be coming out of my mouth shortly. So okay, phytoplankton. Why should we care about phytoplankton? So who, again, someone who's studying phytoplankton in the room, can I pick on Great Lakes work? Why are you interested in phytoplankton? And I'll repeat it for the microscope. Okay. Perfect. Yeah, so we're hearing base of the food web, driving ecological processes. Anyone here studying HABs, harmful algal blooms? Yep. Yes? Try and model HABs. So there's someone in the room say, I'm not sure if it'll pick you up on the microphone, but somebody's saying that they're trying to study HABs, so that's harmful algal blooms. And another one over there as well. So harmful algal blooms is a big one. So I guess going back to more the science and research question, we've been looking at phytoplankton community composition probably more from like a biogeochemical perspective. Looking at how these phytoplankton are driving the carbon cycle, for example. And but as we move forward, obviously there's these things impact our society. Harmful algal blooms closing down beaches, so it's costing you. You go on your nice holiday to the beach, and all of a sudden there's a toxic bloom, and they have to close the beach, and the resort loses lots of money. So there's more than just the biogeochemistry. There's actual human and economic impacts, and if we think about food security as well, even base of the food web, but also if we have a toxic algal bloom that shuts down a fishery, that can be a big problem as well. So we care about phytoplankton in short. And again, just going back, I love these microscope pictures. So this is Zrinka aboard the Zrinka from Croatia. And she's looking down the microscope aboard a ship. It's some different phytoplankton, I presume. And just to give you an idea, again, here's some different types of phytoplanktons. We've got diatoms, dinoflagellates, and showing you how diverse they are. So some of them occur just as single cells. Some of them might be little spherical, you know, like we have Synococcus or Prochlorococcus. They're tiny little things that are very hard to see, very small. And then we have larger diatoms, which can basically stick themselves together and form long chains. And we've got also these dinoflagellates, which is kind of a little bit motile. And so anyhow, different sizes, different morphologies, different colors. So if you think about if things have some different properties that can affect the way light interacts with them, maybe we can start seeing that with our remote sensing. And that's kind of the principle behind all of these phytoplankton community composition algorithms. Okay, so how do we classify? When we say phytoplankton community composition, what are we talking about? In the sort of the contempt of for a while, the terminology we used to use was phytoplankton functional type. And so when I say function, you're kind of thinking what, there's a definition here from the ICCG report. It represents a group of species aggregated according to distinct functional characteristics. So sometimes you might have species that are from, types of plankton from different groups lumped together based on their function in the ecosystem. They're calcifiers, nitrifiers, silicofiers, etc. Another way we would class them or classify them is based on size classes. So we have the people who are not familiar with phytoplankton, you might often hear people talk about micro, pico, nano, and that's microplankton, nanoplankton, picoplankton. And we basically say based on the size, we classify our phytoplankton into these three distinct groups. Another way we might, and we're sort of moving towards now, is grouping more, classifying our community composition by groups. So that might be looking at, we've got diatoms, we've got dinoflagellates, we've got chlorophytes, haptophytes, etc. Splitting it up that way. And another way, which is sort of more maybe for the HABs, I guess, identifications, if we have a dominant group. So something like the coccolithophore that is a white calcium, it's a little tiny plankton that has these calcium carbonate shell, but it reflects light really efficiently. So it turns the water a milky color. So that becomes a dominant bloom that's very easy to see, or something like trichodesmium, which floats at this messy orange scum on the surface. So that's your dominant group. So that there's different ways to, I guess, classify them, and it's kind of up to you, the question you're asking, which one of these ways is correct. Okay, important thing to think about is units. Once we've classified them, how we're reporting the concentrations. We can use absolute units, we can use relative units. Commonly, we will talk about, often we'll say the proportion of the total chlorophyll. So say we have we've taken a bucket of water and we've measured the chlorophyll concentration in that bucket. And then we want to say, well, how much of that chlorophyll was due to diatoms and how much of it was due to dinoflagellates. And so we often will carve up and report our classifications in terms of the proportion of total chlorophyll. That's a common way to do it. And obviously that translates well over to relative units as well. And I'll show you a little plot in a minute, because sometimes relative units help those sort of classifications pop out a little bit more when you're visualizing them. Just say that as well, like probability units of occurrence is quite useful as well. And that's important if maybe you're coming up with a forecast model and you want to say, what's the likelihood or probability that we're going to have this kind of algal bloom coming up based on what we're seeing or what our model says. Okay, so here we've got a comparison of absolute units versus relative units. I just want to sort of, it's hard because it's behind me, in front of me. There it is. Okay, so if you look, it'll be your left-hand side. We've got some phytoplankton groupings based on absolute units. And on the right-hand side, so that's looking at different groups and how much of the total chlorophyll of different groups, but in units of micrograms of chlorophyll per cubic meter. And then on the right-hand side, we're looking at a different model, a different approach, and looking at the relative units. So the proportion in relative terms of the total chlorophyll in percent. And just showing you that, just to sort of give you an idea that visualizing that data it can be done in different ways. Whenever we're reporting data, it's useful to report in both absolute and relative units. That's why I always, because sometimes the percentage of a small number can still be a really small absolute number. So that's just something to keep in the back of your mind when you're looking at a paper. Yep. Okay. So I won't dwell too much on that. Alrighty. There was a paper came out a couple of years ago now called The Consumer's Guide to Satellite Remote Sensing of Multiple Phytoplankton Groups in the Ocean. If this is your game, this is a really good paper to read. It sort of discusses these different ways of classifying phytoplankton with different classification techniques, and then sort of feeds through to what kind of satellite algorithm may be the best choice for you to use. Okay, now, I just wanted to point out before I move on, when you're classifying and categorizing, there's some caveats, right? So a phytoplankton functional type class, for example, doesn't always mean a taxonomic affiliation. For example, as I mentioned before, you might have a type of diatom that straddles two size classes. So it doesn't necessarily mean if you've got a microplankton community, that's all diatoms, for example. It's going to be a mix of other things. And biology's hard. So is anyone familiar with photoacclimation, for example? Can somebody explain to me photoacclimation? So, anyone want to take a stab at it? Yes. Yep. Okay. That's right. They're tricky, aren't they, phytoplankton? Or it can go the other way too, right? If it's a high stress environment, they might turn down the chlorophyll. So basically saying you have to go in understanding a little bit about biology and how they behave. And sometimes you can plug these algorithms in and they might not behave in the way you expect. And you've got to go back and think about some of these physiological responses. Phytoplankton, why are you so difficult? Okay, so going back to a couple of key reviews, I was just sort of glide over these, but just sort of, these are from 2017. So pretty much back then, what they were saying was there were a few gaps which we've sort of been starting to fill in the last few years. Mismatch between satellite and in situ and model data. We're sort of improving upon the way we're validating and collecting field data. I'm going to talk about that soon. Uncertainty is still quite tricky. How do you work out the uncertainty on these measurements? Again, they identified spectral limitations in the existing sensors, not a problem now. We've got things like PACE and up there, and we can do some pretty interesting thing. And sometimes these algorithms are tuned regionally, so they might not work everywhere. And sort of, I guess moving forward, there was another paper as well that said, hey, we should probably do some more of these round robin. Who's heard of these round robin comparison? Anybody? So round robin is where you have a bunch of different algorithms and you want to figure out which one might be the best for your area or how they perform when you compare them to one another. But that can be tricky because sometimes these classification algorithms output different things. So you're not always comparing apples with apples. Anyhow, so we've made some inroads since about 2017 when these papers were published, and things are going in a pretty exciting direction now since PACE has been launched, because that's also driven the community to start collecting more validation data of different types and complementary types all in one go. Which leads me to field measurements. Okay, who's collecting field data at the moment not me. Well, not me, but okay, we've got some field scientists in the room. So that's great. Hopefully I get some things right. If I don't, throw a pen at me. And for those of you who don't collect field measurements, I'll do my best to try and walk you through it. For field measurements for, I guess, quantifying and classifying phytoplankton kind of go back quite a way, and the traditional approach was microscopy. So there was a picture before of someone looking down a microscope, and essentially you throw one of these big nets over the side of the boat, you scoop the water out, or you trap it in one of these Niskin bottles, which are a self-closing bottle, and you take some samples out, you put them under the microscope, and you start counting. And that's tricky. Why? So tricky for a couple of reasons. One, if you're like me and you get really seasick, looking down a microscope on a moving boat, it's not fun. You also have to be really familiar with these phytoplankton keys. So these are big books, or well, not books anymore, but digital, and it shows you the different types of phytoplankton, what they might look like in different orientations. And again, that's another thing that you have to be pretty experienced, because just because what's drawn in the book looks like what it might not be exactly what you're seeing if the phytoplankton has been orientated slightly differently under the microscope. So but it's still bread and butter. People still do it. It's really important to look down the microscope, but there's also some things we can't see under the microscope. The really tiny things like Synococcus, they're just so small. So there's other ways to get to them using things like Coulter counters, and I won't go into that today. So our bread and butter, probably in the last, since CWIF's launch, so the last 25 or 30 years, has been pigment to phytoplankton groups. So we have a technique for looking at, so total chlorophyll A is chlorophyll A is the primary or the main photosynthetic pigment that you have in phytoplankton, but phytos also have these other accessory pigments. So they might assist in light harvesting, or they might be photoprotective. And we have a methodology to look at what those accessory pigments are using high performance liquid chromatography. So essentially you take a water sample, you filter the water through a little funnel onto a glass fiber filter, concentrate it on there, and then you don't have one of these on board a ship, so you have to put it in liquid nitrogen and store it. And then once you get back to the shore, you send it to, I should say this is Crystal Thomas from NASA Goddard here, by the way. Were you wondering why it says 9:55 a.m. on there? Seems a bit weird. NASA was running this, our science goes 24 hours a day. So at 9:55 a.m. on that particular day, Crystal was running HPLC analysis. So anyhow, shout out to Crystal. The data they make in their lab is fantastic. So anyway, once you've got your phytoplankton on your filter, you've shipped it to the lab, frozen in liquid nitrogen, you thaw it out, you put it in a solvent to extract those pigments out, and some people would use a sonicator to kind of break up the cells. In the old days, you'd grind it up, and you put that extract into the liquid chromatography machine. You get these things out called chromatograms, which show these peaks where the different pigments are. And essentially, if you know roughly, you can relate that the relative concentrations of those pigment peaks to different types of phytoplankton. Essentially, you get a big report that says there's this many micrograms per liter of this pigment, there's this many micrograms per liter of this pigment, and you move forward. Okay, so this is the chart. This is one I've stolen from Ali. So credit to Ali. This is a chart basically showing you different types of phytoplankton down the left-hand side. And as you go across, those are the different types of pigments that we're extracting, the names of the different pigments we're extracting with HPLC. Some of them I cannot pronounce, so I will just point at them. And is it that one's a little bit hard to see. Yeah, okay, it is showing up. So you can see there's various gray boxes, and those gray boxes are showing you that those particular pigments are associated or with those particular types of phytoplankton. So essentially, this is just to help you visualize or understand that we should be able to relate different pigment types to different phytoplankton. And if that occurs there, we can come up with a quantitative way to formalize that relationship in some sort of model. Okay, this is a very busy graph. Again, it's very similar to the previous one, and this is from Sasha Kramer. You've got pigments. Pigments are on the y-axis, different types of phytoplankton are on the x-axis, and the color coding is correlation coefficient. So if it's blue, there's a very strongly blue, there's a negative correlation coefficient. If it's red, it's a strong positive correlation coefficient. And again, it's just sort of showing you we can study and understand these different relationships between pigments. And in fact, in this case, we've also got some environmental parameters at the bottom: temperature, salinity, mixed layer depth, light availability, and different plankton. So yes, John. So yeah. Oh, I knew this question would come up. I remember thinking it at the time. Sorry, John. Yeah, no, it is a good question. Yeah, I'll get back to you on that. Yeah, yeah, sorry. I remember thinking that too. I was like, oh, someone's going to ask me this, and I've forgotten. Yeah. Okay. That's where someone gets to throw the pen at me. So throw the pen. So as I mentioned, there's these. We've seen that there are these relationships, whether we can explain them or not, between different phytoplankton pigments and types. And so clever people have come up with these models to relate one to the other. There's two common ones. The first one I'll talk about is called diagnostic pigment analysis. So has anyone used or heard of diagnostic pigment analysis? Okay, so again, if I get it wrong, someone can correct me. So diagnostic pigment analysis, or DPA, takes 7 accessory pigments, and that's the input to your model. And the model tries to output three different phytoplankton size classes, so pico, nano, or micro, with respect to the total chlorophyll. And it's essentially you've got these little these equations down here, and there's these weight coefficients that you have to plug into them. But the weight coefficients are often region specific, and you have to tune them, and you have to potentially be very careful with how you formulate them. Often folks will use cultured phytoplankton, so you know exactly what is in your test tube. If you took some from out in the wild, you're going to have to sort them, and I don't know if that's even possible. And I've truncated this table down on the table of those model weights on the bottom right-hand side because it's actually quite long. So, yeah, that's right, the caveat. So then the second approach we have is chemtax. Anyone heard of chemtax? Chemtax, so chemtax has been around for quite a long time, and the input to this model is these pigment to chlorophyll ratios. So again, you've had some cultures from a laboratory where you know exactly what phytoplankton are in your test tube when you do your HPLC. You do that for a bunch of different types, and then you look at the ratio of the accessory pigments to the total chlorophyll, and you come up with you put it in a matrix, and then you take your measurement in the field, you do the same thing for your measurement in the field, where you look at the pigment ratios, and essentially you do a minimization and inverse model, which is a little bit tricky. What do they? I can't remember exactly what they call it. But essentially you solve for these class abundances, and but instead of looking at size classes, you're looking at classifying different particular groups. Okay, but it's important. There are some caveats. It's going to take a picture of me a minute and blind me. It's okay. But there are some. So we have some caveats, some weaknesses with the DPA and chemtax. So there are some phytoplankton that taxonomic groups, and they share pigments. So maybe it makes it difficult to exactly discriminate between the two. And HPLC doesn't not let's correct me if I'm wrong, but doesn't capture phycobility pigments because they're water soluble often. So, and there are some certain types of cyanobacteria, which that's a sign of having cyanodes as your phycobility pigments. So there is an approach where you can get phyco through your HPLC, but I think there's an extra additional step in the chain. Yeah, there are the groups. It's very distracting. There's the groups encompass you really need to have a priori knowledge of what those accessory pigment ratios are. So you really need to have some high quality cultures that you're taking data from. And what you find is with, for example, like the diagnostic pigment analysis, sometimes you get misclassification. You get microplankton, or you get, say, diatoms straddling the different size classes, and you end up underestimating one size class and overestimating the other. So you kind of need to be careful, and just be aware that most approaches are regionally tuned. So something that a data set that was developed in the North Atlantic probably will not work off the coast of South America, for example. So it's a buyer beware situation. Okay, flow cytometry. So who has used or heard of flow cytometry? Okay, it's the same people coming up again. So flow cytometry it sort of came about from healthcare science. You know, people will analyze blood samples using flow cytometry, and you pour it's a very small sample. It gets drawn out of a test tube, and then a single cell kind of gets injected into what they call a sheath fluid. And that sheath fluid is for what the work we do. You'd filter some seawater through like 0.2 micron filter, so that your sheath fluid has basically the same refractive indices as the natural medium that your plankton would be living in. And you illuminate it goes through a very tiny sort of flow cell, and it gets illuminated by lasers. And you can look at scattering, forward scattering, side scattering, and then fluorescence at different wavelengths. And essentially from that, you can start classifying your plankton samples into different groups. And you get these things called, they're called cytograms. I think they're called cytograms. And you get these kind of scatter plots, and you can start seeing the different groups falling apart. But the caveats with the flow cytometry is the sample sizes are really small. So if you're looking at, like, remember you're trying to say, of this pixel in the ocean that might be under a satellite that's 1 kilometer by kilometer, I put 10 milliliters of water through my flow cytometer. So just think about these kind of things. And if you're in a very clear water, South Pacific gyre or something like that, you've got the particles are very diffuse. So you might have to come up with a strategy to concentrate down. And you've got to carefully calibrate them. But we're all good at calibrating stuff. So flow cytometry leads me to these quantitative cell images, which I think are fantastic. I think it's amazing. So you're kind of taking that concept of flow cytometry, where you're taking a little particle and flowing it through this little channel, but instead you're combining the principles of flow cytometry with microscopy. So now you can actually scatter light and try and get an idea of what's the principles of flow cytometry, but you're seeing it. You can get up here, you get these little microscopic images of what you're seeing in your sample. So that's really cool. So the flow cytometer, the Cytosense and Cytosub, that's a European company, and Imaging Flow Cytobots at McLean Labs in the US. You can combine the flow cytometry and like an imaging microscope. Whereas there's also another instrument out there called a flow cam, but I understand that one's just flow through microscopy. There's no actual lasers scattering things inside there. And I believe the flow cam doesn't pass the particle through in a sheath material. So anyway, these are game changers essentially. These means that if you're collecting HPLC, you've got your imaging flow cytometer, you can start to pull the pigments out, you can start to see what's there. For the really small stuff, if you've got a Coulter counter or something looking at particle size distribution, then hey presto, life's good. Now, with quantitative cell imaging, what's is a lot of data. It's really rich. But that's great because it means you can use these image classification libraries to automate your classification and come up with these machine learning algorithms to do that. And that's pretty powerful because what you can actually do is the machine learning algorithm can start to look at the morphology of the phytoplankton and understand it in different orientations. Obviously, you need to train it properly, but it's very powerful tool. Yeah, with anything, like I said, you've got to have some oversight. You've really got to make sure that what the output if it's telling you it's this type of plankton, you want to every now and again verify. So you got to run some test samples through the machine or whatnot, verification, validation. But anyway, this is a really cool thing. I just put that little cartoon down the bottom just to say, you know, these kind of cloud-based a cloud-based sort of processing system using something like an imaging flow cytometer, you can set up regional monitoring systems. So if all of a sudden you've got this thing sitting out in an estuary and you're looking for a harmful algal bloom, and all of a sudden the system detects it, it might be able to send you a message or send a message to a end user on their smartphone or whatever to alert them to this. So yeah, a lot of cool capabilities. Yeah, that's just giving you an idea of what some real data looks like coming out of one of these systems. So okay, Wayne's world, optical fingerprinting. So essentially, this was a paper that just came out by Mike Lomas, and it's we're calling it optical fingerprinting of phytoplankton. So what it's saying is you're collecting, whether you're doing it in a laboratory or you're collecting it out in the field, you've got a whole suite of optical instrumentation. You've got your list to do your particle size distribution. You've got backscattering meters, you've got absorption and attenuation meters, and you're measuring the full suite of optics and then trying to relate that hyperspectrally to the different type of phytoplankton you're seeing. So just for an example, the bottom right hand, sorry, can I point on here? Can you see this? No. Oops, well, too far. There's this one here. Sorry, everyone. This plot here. So you can see there's a number of different absorption coefficients for different phytoplankton normalized to 1, I think, at 440 nanometers. You can see that those different type of phytoplankton in the absorption space look distinct. So that's important because we'll talk about that with the algorithms in a second. But and also in the backscattering ratio, you can see different phytoplankton have different backscattering efficiencies. Essentially, typically, you know, with me theory, correct me if I'm wrong, Way. Very small things tend to scatter light more efficiently than larger things. So you often will look at if there's things are scattering have a higher backscatter ratio, you tend to think that they might be smaller particles. The bigger particles have the smaller backscatter ratio. That right, Wayne? Yeah, cool. All right. Otherwise, I'm going to say flip it. And another thing I just wanted to point out is that you have to be careful. Again, the caveat here is with measuring what type of measurement you're taking, what instrument you're using. So here we have the blue line on these two sort of plots side by side for two different types of plankton. There is a red line, and that's from a spectrometer, and there's a blue line which is from an ACS meter. And you'll see that some of the ACS misses some of these spectral features, and that's just due to the design of the AC meter. It's got some the bandpass on the filters tends to degrade some of these spectral features. Just it's harder to resolve them. So just a thought. Okay, and then finally, something I'm not too familiar with, but it's becoming more and more popular, is the DNA meta barcoding. So essentially, you can take a sample of water, run it through a DNA analyzer, and some people are doing research to link what DNA barcodes are linked to certain pigments or certain phytoplankton. And so that's becoming a bigger and bigger now in this world of phytoplankton community structure. Okay, so just finally, before I move on to satellites, just thinking about a few thoughts. You got to think about designing your study. What do you need to collect? Because it's expensive and it takes people and it might take a boat, budget, time. And each approach that we've described for field measurements has its strengths and weaknesses. In an ideal world, you'd try and collect and measure all the data at the same time, but that's often in practice very, very hard to do. And one thing is, it's important to try and think about how you might quantify measurement uncertainty. John talked about measurement uncertainty before, and it's a tricky thing because we sometimes just want to go and take our measurement and move on to the next thing. But characterizing uncertainty is really important, especially if you're validating a satellite algorithm, for example. So, oh yeah, and I chucked this in here because this is, sorry, I shouldn't say I just chucked this in here. What I mean is this is a very important to think about, the FAIR principles. And, it really what we're saying is if you're going out and collecting data or creating satellite algorithms that we want to try and, NASA especially is trying to adhere to these, the FAIR principles: findable, accessible, interoperable, reusable. So making your data available to others, making sure that it's well documented and it's consistent, other people can get it and use it if they want it. And that helps feed this open science environment, and which we all like the open science. Sharing code on GitHub, if there's a bug or something, people can get back to us, we can fix it, that sort of thing. So making it more democratized, so people people might be developing an algorithm, but they can't afford to buy the instrumentation that you have. So you're sharing the data with other people and creating this community that innovates and moves forward. Okay, all right, I'll take a very, very short break here. This is a quiz to see what phytoplankton you are while I take a drink of water. Sometimes this doesn't work straight away, so you got to hit refresh on the browser, and we can talk. Is it working? It's spinning. I think you have to hit refresh on it. I've had that problem before. Good, yeah. It'll ask you a few questions. No, this is not a marketing scam where you'll start getting advertisements for baseball hats or something. Do you get money? No, I get money. No, just kidding. I can't remember how many questions there are. I think there's only about 5 questions. Has anyone got an answer yet? Prochlorococcus, like it. Yeah, not bad. Yeah, nice and trichodesmium, like it. Okay, yeah, there we go. One up there. Dinophysis. Can you read out the description of what dinophysis is? Nice. And what was the tricho one? Wayne, what was yours? Well, so we're in a warm place now, so yeah. Okay, well, I'll move on now, but just a bit of fun, so helps me rest my voice. Anyway, John, what did you get before we move on? Are you a coccolithophore? Dinophysis. Fun fact about dinophysis, which they don't put in there, is I recall that that's one that can give you a really bad upset tummy if it creates a toxic.
It's a seratium.
What was that?
Seratium.
Ceridium. Excellent. Okay, cool, fun. Let's move on. And you can share that and play with that later. Okay, let's talk about let's talk about satellite measurements and scaling up to remote sensing. I'll try and get through this. So this is a picture from a paper by some guy looking at trichodesmium and sort of just showing you at different length scales what trichodesmium looks like. So under the microscope, it can occur as single cells or filaments. You're the expert now on trichodesmium, or it can form these macroscopic colonies. You can actually scoop out the bloom water on your hand and you can see the colonies without a microscope. It's pretty impressive. And when it blooms on the surface, you tend to get this really brown, stinky munge. And then when you zoom out to outer space, you can see it lights up hundreds of thousands of kilometers, sometimes, these floating blooms. And it's a really, really important, especially in the oligotrophic parts of the world, because they're nitrogen poor, and so they actually fix atmospheric nitrogen and bring new nitrogen into the system. So just to give you an idea, microscope to outer space. So doing this from outer space, detecting phytoplankton from outer space, we kind of have two main approaches. We can break them down into abundance approaches and what we call spectral approaches. And if we go back to that sort of classification chart I showed earlier, from the consumer's guide to satellite remote sensing of plankton groups, we can start to break up the approaches for classifying phytoplankton in terms of these two remote sensing pathways. So the abundance methods is basically you're given some sort of measure of phytoplankton biomass, whether that's the chlorophyll concentration or the magnitude of the phytoplankton absorption coefficient, which you derive through the inverse models, which I think we were talking about yesterday's lab. And then you use that information, but the magnitude of that information helps inform you about what type of phytoplankton size class you may be viewing from space. So this is I call this a spaghetti diagram. And so if we're talking about abundance-based methods, we're looking at these red pathways from satellite-derived chlorophyll, for example, down to size class or PFT taxonomy. So I'll give you an example. This one's by Taka Hirata and a few other people who we know. And the input to this model was total chlorophyll concentration. And essentially, you use this pre-existing knowledge of what size classes you expect to occur given a particular a particular chlorophyll concentration. So if you're in think of it this way, if you're in very, very low nutrient oligotrophic waters where the chlorophyll value is small, you tend to have very small phytoplankton. But if you're in very, very rich, well-fertilized, let's say coastal upwelling, and you've got a lot of food for things to grow, you tend to have a lot of larger phytoplankton. And if you're somewhere in between, you've got the nanoplankton. And so when you put this into a satellite algorithm, you tend to get these plots that fall out. And as you can see, the areas, let's just look at that first row, we've got micro on the left-hand side, nanoplankton, then picoplankton. Just going back to that example of low chlorophyll waters, you can see the areas that are lit up red. Everyone see those? In the picoplankton. So that's areas where we have very, very low nutrients, very low chlorophyll, and so we assume that there's a lot of picoplankton, little tiny things. We can also, instead of taking chlorophyll to get to our size class, we can take the magnitude of the derived phytoplankton, or the magnitude of the derived absorption coefficient. So in this model, they were looking at the shapes of different phytoplankton based on their size class in that panel. And you can see that between the microplankton, which is the red, the nano and the green, and the micro, pico's blue, nano's green, micro's red, that the magnitude and the actual slope of that absorption coefficient changes between, say, 450 and 500 nanometers. So you can essentially use that information to inform you about your size classes that might be present. And they basically came up with these thresholds to say when the phytoplankton absorption coefficient that we've derived from the satellite meets these thresholds, we classify our plankton in these particular ways. So there's so the abundance methods, you really, really need to have an understanding before you I guess run the model. You need to understand those distributions of phytoplankton size classes in the ocean and how they relate to chlorophyll concentration or the phytoplankton absorption coefficient. The other approach, the other pathway, is what we call the spectral methods. So instead of looking at just one parameter, you tend to look at the shape, whether it's the shape of the reflectances or the radiances, or even the inherent optical properties that you've derived. And it allows us to step beyond phytoplankton size classes into more of the PFT or taking different groups out. Right, what else do we say there? Okay, so let's look at this pathway where you go from satellite measured radiances. So do we talk about radiant? Everyone familiar if we're talking about radiances, what I mean? So it's basically it's like, yeah, reflectances, but it's not normalized by downwelling irradiance. So if we look at, for example, here, this was just a very simple sort of empirical model looking at the pigment peridion. That's on the y-axis here versus a ratio of, so again, looking at spectral shape, I know that's a band ratio between 490 and 670. And also we can look at, for example, harmful algal bloom in the Great Lakes through the Cyan project. This is another example where we're looking at the spectral shape. In this case, we're looking at a red edge reflectance where we have basically a near surface floating harmful algal bloom, and that changes the spectral shape. But we need to look at it collectively. We can't just look at the magnitude of one value to understand if we have a bloom present. But that one's called the cyano index. So this is a good one. This was when they started. People were Severin Alvain and a few others were looking at how the shape of the reflectance normalized to something, whether it was chlorophyll. It was chlorophyll case here. So you basically build up a library of phytoplankton. Needed to give you guys a longer break for my brain. You look at the normalized water leaving reflectance shape, and then you normalize that by chlorophyll concentration. And you kind of build up a library of what you expect that shape to look like based on different phytoplankton groups. And then you kind of, when you run your model, you have a lookup table where you match closest one of these, I think they ended up calling it like a radiance anomaly spectrum with, from your lookup table with what you're seeing in your satellite imagery. But the idea there was to just look at what is the dominant phytoplankton type in that particular pixel. It wasn't looking at grouping and proportions. It was the dominant one. Okay. Next pathway on the spaghetti diagram, going from total absorption down to accessory pigments and phytoplankton community composition. So we could use the particle absorption coefficient or the phytoplankton absorption coefficient as an input. This was work by Dylan Catlett and Dave Seagal, and essentially they were looking at the derivative spectrum. So has anyone worked with derivative spectroscopy before? Yes? Okay, so you can tell us what are you trying to do with what is the derivative doing to the spectrum when you apply it? Right, no, it's right, yes. It helps, it can help the noise. If the noise is too big, that can also exacerbate the noise and you get a false signal. The other thing it helps do is it kind of normalizes. You're normalizing to you got 0 and then you've got these spikes either side. But essentially, yeah, I'll say it for the recording. So just what the comment was, or the answer was, is you perform a derivative upon a spectrum and it essentially pulls out these, there may be sometimes subtle features that are associated with a pigment that's a fingerprint for a certain phytoplankton. So that's one pathway when you're looking at total absorption. Another pathway developed by Alice and Chase is you try and create a model based on a bunch of Gaussian bands, which are then related to different pigment concentrations. And you run this and you run essentially like a spectral matching until you get your observed absorption spectrum matching with your Gaussian model. And the magnitude of those different Gaussians and where they're spectrally placed and their width tells you something about what pigments are present. Okay, then we're going back to the spaghetti diagram. We're going from satellite measured radiances and derived IOPs and this pathway here. So this is an approach that Jeremy came up with. And essentially, so you guys were working on the inverse model exercise yesterday, is that right? So and a bit like John was talking today about this, you have these models where you're doing spectral matching. You're trying to have a forward model that's trying to generate a remote sensing reflectance, and you tweak all the parameters to try and match what the satellite's seeing with what your model sees. And in this, typically in contemporary satellite remote sensing algorithms, we would maybe just have one phytoplankton shape because we're only working in multispectral worlds. So there's not too much difference between five bands of noctiluca and 5 bands of diatoms. But in this case, Jeremy put in a mixing model. So you're trying to figure out what was the dominant phytoplankton in this from a spectral matching algorithm by putting in a shape for diatoms and a shape for noctiluca. And similarly, there was again, instead of going from the species level down to the size class, Aurea Schotti came up with a phytoplankton absorption shape model, which was a function of essentially a linear mixing model between two different size classes of phytoplankton, and then based on what the answer that you got out for this, what's called the SF, the scaling factor, it will give you an idea of the relative proportions of these two size classes. Okay. Yes, and there's another way we can get from satellite-derived IOPs to size classes, and that's through looking at the backscattering coefficient. So Tiho Kostadinov came up with an approach. It's sort of based on this idea. I talked a bit earlier about the scattering properties can tell you a little bit about size class, and that previously I was talking about the backscatter ratio. But if you look at the slope of the backscattering coefficient, that can also give you an insight potentially into the size of the things that are in the water column. So let's think about, are you guys familiar with what the shape of the backscattering coefficient usually looks like when we model? No. Have we got a whiteboard I can draw on? Yeah, sorry, it's not going to come up on video, but so when we're looking at the backscattering coefficient, we usually use like a power law model, lambda on some reference wavelength, some power exponent, and then I think in John's model, it was an X, wasn't it, for the magnitude of. So we're going to have this is backscattering coefficient, BBP. Sorry, I didn't plan to do this, so I can draw it on there, can I? Oh. Cool. Okay. Okay. So essentially, your backscattering coefficient spectrally. Oh wow! Yeah, cool. Sorry, this is fantastic. So you have a backscattering coefficient that is sort of, in reality, it has wiggles and bumps in it, but for most intents and purposes, in the past, again, we're living in multispectral world, we typically model it with a power law. But this exponent here in your power model can tell you a little bit about size class. So sometimes you might get a very flat backscattering coefficient, or you might get a very steep backscattering coefficient, and that's driven by this exponent here. So when it's very, very steep, that's often an indicator that you've got small things, and when it's flat or goes that way, it's often an indicator that you've got bigger things. So that's essentially the principle of Theo's model here. Okay, that was fun. I've got to get back to your share PC. No.
How you connect? Where did you connect the pin drive?
No, it's just connected to there.
No. No. Yeah.
Aha. Okay, good. It's just gone to sleep. Okay, so so what you're seeing down. We're done. Wake up. No. It's gone. It's back. It's gone. It's back. Don't touch anything, Lachlan. Okay. So what you see there's a global map here of what we assume or what we can model or derive the spectral the slope of the spectral backscattering coefficient. So you see where it's bigger. The number's bigger is again in these oligotrophic gyres in the central parts of the ocean where they're very nutrient limited and we have lots of small stuff. And then it's the slope gets bigger where we have more productive waters and we tend to have bigger things in general. So TIHO basically used this principle with some modeling to come up with particle size distribution slope, and then you can link particle size distribution to phytoplankton size classes. Okay, on to the hyperspectral era. So I'll talk a little bit more about this tomorrow, and I think this plot, maybe this has showed up a few times. We're living in the hyperspectral era. We've got a 2-day global coverage orbit with PACE, 1 kilometer pixel. So we're stepping on beyond where we were with MODIS, and we can start looking at more of these spectral inputs. There's been some really nice reviews recently by Heidi Dierson, Ivona Setnich, and a bunch of people, I think my name's in there somewhere, about where we're moving to with these phytoplankton community composition algorithms in the PACE era. Derivative spectroscopy is one very, very exciting avenue. It was sort of proved by Astrid Bracher and a few others quite a few years ago now using Sciamarci data. That was not an ocean color mission, but it had a very, very fine spatial resolution. I think it might have been trace gases, or it was an atmospheric mission. And more recently, Sasha Kramer with her, again, it's a spectral derivative. And going back to what we were talking about before, if we look at the derivative of the remote sensing reflectance, you start to see you can really, really tease out these fine scale features when you perform a derivative analysis. Machine learning is another really exciting area where we can start, taking this. Again, you have to build your model. And I know there's some machine learning people here. Yeah. Okay. Yeah. Good. So machine learning people, you want to have a very, very good training data set. So whether you use John's hydrolite to build that, or you collect field measurements, you come up with this really high quality sort of machine learning process and perform some feature extraction or feature reduction often, and then you can push those models, those variables that might explain more, parameters that explain, for example, a principal component, which explains 90% of your variability, can go into your model. And, depends what your model is. It might be a principal component regression, neural network, etc. Oh yeah, and I was going to say something. Sometimes, we forget that we live in the oceanography or bubble, that people do this all the time in the outside world. So this sort of automated imaging for automated imaging using neural networks and hyperspectral little tiny cameras is really common in things like the food industry. So they have, is there mold on this piece of cheese that's going down to the production line? And if there is, it gets kicked out. Or they're scanning fruit, and has the fruit got a blemish on it? These are kind of, the concepts that we use outside. And that's kind of also those compact spectrometers that Wayne was talking about yesterday. A lot of them are for process control in manufacturing and food science. So we just take the technology and use it for science once they figured it out. And I just wanted to, there's a sort of a model here that was developed by Lange et al. A couple of years ago, and that was looking at doing a principal component analysis and classifying phytoplankton using, it was actually with MODIS data, but we can scale it up to PACE. Okay, now for those of you, I think I've gone through this just then, but with machine learning, we want good data. We need to train it carefully, don't we? Here's an interesting thing. So for example, with the principal component models, there was one of my colleagues was saying that, often we take the principal components that explain the highest degree of variability. You do these scree, it's called a scree plot, and you've done your dimension reduction, and these 4 principal components explain 99% of the variability. But sometimes they're not the best predictors. Sometimes there's a predictor that explains a very small proportion of the variance that actually works really well. So you have to think carefully about how you're selecting the inputs to your model in a stepwise approach or whatever you choose to do. And also, these things need regional training, may need regional tuning, don't necessarily work globally. But the good thing is they can be really robust. If you train a neural network to expect noise or some sort of weird thing in your data, they can be really robust. And what's really cool is if you use some sort of radio transfer modeling, for example, you can train your neural network or machine learning algorithm with top of atmosphere radiances. So you don't even need to do an atmospheric correction. You could just take calibrated top of atmosphere radiances from PACE and just push it through to get your retrieval. But people have shown to do that as well. I think Suzanne Craig is working on that. All right, part 4, algorithm validation. Try and skip through this quickly. What's the general approach? Who's done validation here before where we're what's the purpose of validation? I should start with, actually. Have we discussed algorithm validation during the last week and a bit? I'll take that as a no. Sorry, John, I did. John's sitting in the background, you jerk. You weren't listening to my talk. Sorry, John. But essentially, it depends what you're validating, but the purpose is validation. Meteorologists call it forecast verification, but essentially you want to make sure that you take a measurement in the field, compare it to what your satellite's seeing. In John's case, looking at bathymetry, in this case, looking at a phytoplankton group or something, and make sure that the satellite is performing as expected. And this is sort of the general workflow that NASA has come up with and been using for a few years to validate their algorithms. Essentially you take your satellite pixel, you do some quality control, and you compare it with your field measurement. And in the past, we'd make these one-to-one scatter plots, linear regression plots, and report things like slope, intercept, R squared, and that can be problematic. So one, type 2 linear regression should really only be used on normally distributed data. So we tend to do these log transformations of all our oceanographic data, because normally in the ocean things follow a skewed distribution. But for example, like, what about this? I'll just give you a question. What do you think about what I'm showing you here? This is a one-to-one scatter plot. So if you're going by the principle of a one-to-one type 2 linear regression, if it's a good retrieval, theoretically, all your data points should fall along that one-to-one plot, right? That's what we're looking for. But in this case, we're looking at the remote sensing reflectance at 670 nanometers in very, very clear water out of Hawaii. There is no dynamic range out there. So you don't get a one-to-one line. What you're seeing, that vertical variability, is actually variability in your retrieval, the uncertainty in your retrieval. So you have to be careful thinking about regression metrics. Instead, what we're starting to try and do is move towards what we call this mean absolute error, mean absolute bias. Just think about pairwise comparisons. You've got two pairs of data in situ field, and you want to measure them and essentially just look at the residuals. In this case, we've started plotting a lot of our data using these bland Altman plots. Again, something we borrowed from, I think, the medical research from many years ago. And what this lets you see is also if there's any scale dependent bias, like maybe you're retrieving really well in the low end, but all of a sudden it just kicks up in the higher dynamic range. So just think about, be careful visualizing. This one is called, for people who are doing classifications. So you might, instead of using continuous data, you might be using categorical data. And these things are called confusion matrix. Ideally, what it helps you understand is if you're getting false positives or false negative retrievals, and ideally you want all your answers to fall on the diagonal. But that's something to think about depending on what algorithm you're developing. Okay. Yes, there's been a recent review on this for phytoplankton. I won't spend too much time on that. And it is important to understand this is another paper by Sasha. If you're using what data source you're using for validation, and do they agree? So for example, here at the top row is actually looking at like a diagnostic pigments or HPLC retrievals, and down the bottom you're looking there at a DNA sequence. Just you got to make sure that if you're using these data sets together, are they agreeing? Are they different types of field measurements that you're using for validating? Are they telling you the same thing? Sometimes they might be a bit different, and that causes which one do you choose. So, okay, that's what I look like on a daily basis now with my bald Homer Simpson head. Just thinking finally as well, what if you've got multiple algorithms and you want to compare them? So we've talked a lot about the different approaches that and where we're headed in the future. And you might say, I want to pick an algorithm for this particular location, but I don't actually know which one to pick. How do I go about doing that? So this is the paper by Bob Bruin quite a few years ago now, and this was for chlorophyll, for example. But he sort of said there's ways you can compare algorithms and look at how they perform and basically tally up like wins, and eventually you get an overall score of how the algorithms perform relative to one another, and that can help guide your selection for where you want, for what you're looking at. Or sometimes in the computational environment we're in now, maybe you just want to run all your algorithms together and come up with some sort of aggregate at the end. It's again, it's a question. Okay, so in conclusion, we have many different in situ measurement approaches now that exist. It's really exciting. You know, microscopy, HPLC, cytometry is sort of the historical bread and butter, and we're moving into these new imaging techniques as well as DNA. So if you can sort of measure them collectively, it can help you paint a fuller picture. We sort of can split our historical satellite algorithms into two approaches. We've got these abundance methods and spectral methods. But as we're moving in the hyperspectral era, obviously there's a lot we can do with machine learning, derivative spectroscopy, and sort of, I think we come back and we've sat in the same room in another 10 years' time. I couldn't actually predict what we're going to see. It's a really exciting time. And validation, yeah. So when we're doing validation, you're comparing things together. Think carefully about how you're doing it, using appropriate metrics. And yeah, think about things to think about. If you're trying to select an algorithm, you've got to come up with a way of comparing them appropriately. And uncertainties. I didn't talk about uncertainties. I was going to, but I figured I'd run out of time, and I have. But uncertainties in your measurements are a thing to consider, because when you're comparing, data point A with data point B, really every measurement has a probability density function around. It's got an uncertainty. So those probability density functions, if they're really big, you've got a lot of uncertainty in your measurement. They might actually be too large to statistically discern a difference between your two data points. So I can talk more about that another time. But anyhow, thank you. Keep sorting phytoplankton.